Hi class, this is your instructor, Skylar Huff, and we're now in Chapter 7, How Cells Harvest Energy from Food. Let us begin. So where is the energy in food? You will distinguish between oxidation and reduction, which I always call redox reactions. In plants and animals, and in fact in almost all organisms, the energy for living is obtained by breaking down the organic molecules originally produced by photosynthetic organisms, such as plants, algae, and again I'll say certain bacteria. The energy invested in building the organic molecules is retrieved by stripping away the energetic electrons and using them to make ATP, a process called cellular respiration. Do not you dare confuse the term cellular respiration with breathing of oxygen gas that your lungs carry out, which is simply respiration. So this is not respiring, this is cellular respiration. The cells of plants fuel their activities with sugars and other molecules that they produce through photosynthesis and break down in cellular respiration. Non photosynthetic organisms eat plants extracting energy from plant tissue using cellular respiration. Excuse me. Other animals, like the child you see in figure 7.1 on page 120, gobbling up the hamburger, eat these animals. Redox reactions. Eukaryotes produce the majority of their ATP by harvesting electrons from chemical bonds of the food molecule glucose. The electrons are transferred along the electron transport chain, similar to the electron transport system I mentioned in the last chapter, and eventually donated to oxygen gas. Chemically, there is little difference between this process in a cell and the burning of wood in a fireplace. In both instances, the reactants are carbohydrates and oxygen, and the products are carbon dioxide, water, and energy. You see that now. So this is more or less photosynthesis in reverse. C6H12O6 plus 6CO2 yields 6CO2 plus 6H2O plus energy. In a chemical reaction, when an atom or molecule loses an electron, it is said to be oxidized, and the process by which this occurs is called oxidation. The name reflects the fact that in biological systems, oxygen, which attracts electrons strongly, is the most common electron acceptor. This is certainly the case in cellular respiration, where oxygen is the final electron acceptor. Conversely, when an atom or molecule gains an electron, it is said to be reduced. The process is reduction. So oxidation and reduction always take place together because every electron that is lost by an atom through oxidation is gained by some other atom through reduction. Therefore, chemical reactions of this sort are called oxidation reduction reactions, or simply redox reactions. So in redox reactions, energy follows the electron as shown in figure 7.2 on page 120. So if you look there, you see A plus B, and then you see the movement of the electrons. So A loses the electron, becoming oxidized, whereas B gains an electron, becoming reduced. Let's get back to cellular respiration. Here we go. Cellular respiration is carried out in two stages. You can see this very same in figure 7.3 of your textbook. And I say this very same, meaning it shows the entire process on page 120 in both plant and animal cells. The first stage uses coupled reactions to make ATP. This stage, called glycolysis, takes place in the cell's cytoplasm. Importantly, it's an anaerobic process requiring no energy. This ancient energy extracting process is thought to have evolved over 2 billion years ago when there was no oxygen in the Earth's atmosphere. The second stage is aerobic, requiring oxygen, and takes place within the mitochondrion. The focal point of this stage is the Krebs cycle, a cycle of chemical reactions that harvests electrons from carbon and hydrogen chemical bonds and passes the energy-rich electrons to the electron carrier molecules called NADH and FADH2. These molecules deliver electrons to an electron transport chain. And as they do such, the electron transport chain uses their energy to power the production of ATP production. The harvesting of electrons, a form of oxidation, is far more powerful than glycolysis at recovering energy from food molecules, and is how the bulk of energy used by eukaryotes, including me and you, is extracted from food molecules. Now let's get into the process, and let's use coupled reactions to make ATP. The first stage of cellular respiration takes place by way of a series of 10 reactions called glycolysis. I won't ask you to list all 10 of these things, but let's get how the action, or at least the reactions, work. You will see this essential biological process, 7a, at the bottom of page 122. So by way of glycolysis, this 6-carbon sugar glucose is cleaved into 2 3-carbon molecules of pyruvate. Where is the energy extracted? Well, in each of the 2 coupled reactions, the breaking of a chemical bond releases enough energy to drive the formation of an ATP molecule from ADP. That's an endergonic reaction. This transfer of a high-energy phosphate from a group of a substrate to ADP is called substrate-level phosphorylation. In the absence of oxygen, this is the only way organisms can get energy from food, and that is shown here now from left to right. First, it's primed at number 1. Cleavage, or at least splitting, happens in number 2, and then ultimately ATP is synthesized in number 3 from left to right. Harvesting electrons from chemical bonds. So here you will name and describe the enzyme that removes CO2 from pyruvate. The first step of oxidative respiration in the mitochondrion is the oxidation of the 3-carbon molecule called pyruvate, which is the end product of glycolysis. The cells harvests electrons from pyruvate in 2 steps. First, by oxidizing pyruvate from acetyl-CoA, then oxidizing acetyl-CoA in the Krebs cycle. Pyruvate is oxidized in a single reaction that cleaves off one of pyruvate's 3 carbons. Pyruvate dehydrogenase is a complex of enzymes that removes CO2 from pyruvate and is one of the largest enzymes known. It contains 60 subunits. By way of this reaction, a hydrogen and electrons are removed from pyruvate and donate to NAD+ to form NADH. Now, if you look closely, or by way of figure 7.4 on page 123, so the 2-carbon fragment called an acetyl group that remains after removing CO2 from pyruvate still contains a great deal of energy. So it is joined with a cofactor called coenzyme A, or CoA, by pyruvate dehydrogenase, forming a compound known as acetyl-CoA. So if a cell has a plentiful supply of ATP, acetyl-CoA is funneled into fat synthesis, so with its energetic electrons preserved for later needs. If a cell needs ATP now, the fragment is directed instead to the Krebs cycle, where its energy is harvested to make ATP. So now let's keep on going to get into what is called the Krebs cycle. Here you will identify the overall substrates for the 9-reaction Krebs cycle and overall products. And again, you will not go through all 9 reactions. So the Krebs cycle, which occurs hereafter in oxidative respiration, was named after the person, the scientist, who discovered it. So it harvests energy of acetyl-CoA fragments. So the Krebs cycle reactions take place within the mitochondrion. Let's continue. The first step, the cycle starts when a 2-carbon acetyl-CoA fragment produced from pyruvate is stuck to a 4-carbon sugar, producing a 6-carbon molecule. Then, in rapid-fire order, 2 carbons are removed as CO2. The electrons are donated to NAD+. The 4-carbon molecule is left. A molecule of ATP is also produced. And lastly, when it is all over, 2 carbon atoms have been expelled as CO2. And then more energetic electrons are extracted and taken away as NADH or FADH2, and we're left with that same 4-carbon sugar that we started with. More or less, you can see the same here, but I will not have you drawing this out or explaining this on your test. Acetyl-CoA comes in, CoA is removed, the 6-carbon molecule, then we get NADH, we get another NADH, CO2 is removed, we get ATP. Then the cycle starts over again because we now have not just NADH here, but we now also have FADH2. This is shown to you on page 124. So the process is a cycle or a circle of 9 reactions. So in each turn of the cycle, a new acetyl group replaces the 2 CO2 molecules that were lost, and more electrons are extracted. So in the process of cellular respiration, glucose is entirely consumed. All that is left to mark the passing of the glucose molecule into 6 CO2 molecules is its energy, and it's preserved in 4 ATP molecules. And then the electrons are carried by 10 NADH and 2 FADH2 carriers. Now let's get to making the electrons to make ATP. So electrons carry energy from one part of the cell to another. In eukaryotes, aerobic respiration takes place within the mitochondrion, and is present in virtually all cells. So the internal compartment, or the matrix of the mitochondrion, contains the enzymes that carry out the reactions of the Krebs cycle. The electrons harvested by oxidative respiration in the matrix are then passed along the electron transport chain, and the energy they release transports protons out of the matrix and into the intermembrane space. I'm showing you this to allow you to see what I'm saying. The NADH and FADH2 molecules formed during the first stages of the aerobic respiration each contain electrons and hydrogens that were gained when NAD plus and FAD were reduced, and we just saw that. So it's now NADH and FADH2 molecules that carry their electrons to the inner mitochondrial membrane, where they transfer the electrons to a series of membrane-associated molecules collectively called the electron transport chain, which you can see now in figure 7.6 on page 125. The electron transport chain works much like the electron transport system you encounter in studying in photosynthesis. So the first thing that happens is harvesting the electrons. So a protein complex, which is that light pink you see, receives the electrons using a mobile carrier, and it passes these electrons to a second protein complex, which is the purple structure that is adjacent. This protein complex, along with others in the chain, operates as a proton pump. Operates as a proton pump. So it's using the energy of the electrons to drive the proton out across the membrane into the intermembrane space. We're creating a gradient here. The arrows indicate the transport of protons into the top half of the figure, the intermembrane space, creating this proton gradient. The electron is then shuttled by another electron carrier to the third protein in the complex, that is a light blue. And this complex uses the electrons, such as this one, to link oxygen atoms with hydrogen ions to form molecules of water. It is the availability and plentiful supply of oxygen, the electron-accepting molecule, that makes oxidative respiration even possible. So the electron transport chain used in aerobic respiration is similar to and may well have evolved from the electron transport system employed in photosynthesis. So photosynthesis is thought to have preceded cellular respiration and evolution of the biochemical pathways, generating oxygen that is necessary for the electron acceptor in cellular respiration. So natural selection didn't start from scratch and design a new biochemical pathway for cellular respiration. Instead, it built on the photosynthetic pathway that already existed, which uses many of the same reactions. So let's now make ATP. As the proton gradient in the intermembrane space rises above that in the matrix, the concentration gradient induces the protons to re-enter the matrix by diffusion through a special proton channel called ATP synthase. So it is ATP synthase channels that are embedded in the inner mitochondrial membrane, shown in this very same illustration, as you see there at the very bottom right-hand side near number 4. So as the protons pass through, these channels synthesize ATP from ADP and that inorganic phosphate within the matrix. So the ATP is then transported by facilitated diffusion out of the mitochondrion into the cell cytoplasm. This ATP synthesizing process is the same chemiosmosis process that you encountered in studying photosynthesis. So now, how many ATPs can one glucose yield? Although we have discussed the electron transport and chemiosmosis as separate processes, in a cell they are integrated, as shown in figure 7.6. Hopefully you all do notice that. So the electron transport chain uses 2 electrons harvested in glycolysis, 2 harvested in pyruvate oxidation, and 8 harvested in aerobic respiration to pump a large number of protons out across the inner mitochondrial membrane. Their subsequent reentry back into the mitochondrial matrix drives the synthesis of 34 ATP molecules by chemiosmosis. And then there are 2 additional ATPs that were harvested by a coupled reaction in glycolysis, and 2 more in the Krebs cycle. So as 2 ATPs must be expended to transport NADH into the mitochondria by active transport, the grand total of ATPs harvested is thus 36 ATP molecules. So now, cells can metabolize food without oxygen. So you're now going to distinguish between ethanol fermentation and lactic acid fermentation. So electrons on the move, they always find a home. In the absence of oxygen, aerobic metabolism cannot occur, and cells must exclusively rely on glycolysis to produce ATP. And under these conditions, the hydrogen atoms and electrons that were involved in the oxidation of NAD plus and NADH in glycolysis are now donated to organic molecules instead of the electron transport chain in a process called fermentation. So fermentation recycles NAD plus so that glycolysis can continue. We'll be here firstly at ethanol fermentation. Bacteria carry out more than a dozen kinds of fermentations, all using some form of organic molecule to accept the hydrogen atom from NADH. By contrast, eukaryotic cells are capable of only a few types of fermentation. In one type, which occurs in single-celled fungi called yeasts, the molecule that accepts hydrogen from NADH is derived from pyruvate, and the end product, meaning the end product of glycolysis itself. So yeast enzymes removed a CO2 group from pyruvate through decarboxylation, producing a 2-carbon molecule called acetaldehyde. So that's acetaldehyde. The CO2 release causes bread made with yeast to rise, whereas bread made without yeast, called unleavened bread, does not rise. The acetaldehyde accepts a hydrogen ion from NADH, producing NAD plus and ethanol. So if you look at figure 7.8, that is on page 128, you will see this itself with ethanol fermentation in yeasts. This particular type of fermentation is of greatest, I'm sorry, this particular type of fermentation is of great interest to humans, not greatest interest, because it is a source of ethanol in wine and beer. So ethanol is a byproduct of fermentation that is actually toxic to yeast. As it approaches a concentration of about 12%, it begins to kill the yeast. That is why naturally fermented wine contains only about 12% ethanol. Lactic acid fermentation in most animals regenerates NAD plus by a second type of fermentation. So using the enzyme called lactate dehydrogenase to transfer a hydrogen atom from NADH back to pyruvate that is being produced by glycolysis. So it's this reaction that converts pyruvate into lactic acid and regenerates NAD plus from NADH. So this is also in figure 7.8, however it's at the bottom with lactic acid fermentation in muscle cells. It therefore closes the metabolic circle, allowing glycolysis to continue as long as glucose is available. Circulating blood removes excess lactate from muscles, and that's an ionized form of lactic acid. So it was once thought that during strenuous exercise, when the removal of lactic acid could not keep up with the production, the accumulation induces muscle fatigue. However, scientists now believe that lactic acid is actually used by muscles as another source of food. So there you have it. Ethanol fermentation and lactic acid fermentation. So glucose is not the only food molecule. Yes, it is a food molecule, just not the only one. So you'll describe how cells garner energy from fats and then proteins. We have considered in detail the fate of a molecule of glucose, a simple sugar in cellular respiration. But how much of what you eat is sugar? As a more realistic example of the food you eat, consider the fate of a fast food hamburger. The hamburger is composed primarily of carbohydrates, fats, and proteins. So this diverse collection of complex molecules is broken down by the process of digestion in your stomach and intestines into simpler molecules. So carbohydrates are broken down into simple sugars, such as glucose, fats into fatty acids, and then proteins into amino acids. So nucleic acids are also present in the food you eat, but these macromolecules store little energy that actually is used by your body or mine. Cellular respiration of fat. I'll do fat before protein because those three metabolic routes our body takes are in this order. First, carbohydrates, second, the lipids, and then thirdly, proteins. Lipids and fats. They are broken down into fatty acids, and then the fatty acid typically has a long tail of 16 or more carbon-hydrogen links, and many carbon-hydrogen bonds in these long tails, which provide a rich harvest of energy. Enzymes in the matrix of the mitochondrion first remove two acyl groups from the fatty acid tail, and then another, and in effect, chewing down the length of the tail in two carbon bites. Eventually, the entire fatty acid tail is converted into acetyl groups, and each of the acetyl groups then combines with coenzyme A to form acetyl-CoA, which feeds into the Krebs cycle by way of beta oxidation. Proteins, that third metabolic route after lipids and after the first of three being carbohydrates. So proteins are broken down into their individual amino acids by way of deamination. So a series of deamination reactions removes the nitrogen side groups called the amine groups or the amino groups, and it then converts the rest of the amino acid into a molecule that takes part in the Krebs cycle. For example, alanine is converted into pyruvate, glutamate into alpha-ketoglutarate, and aspartate into oxaloacetate. So the reactions of the Krebs cycle then extract high-energy electrons from these molecules and put them to work making ATP, as you see before yourselves there. So this is figure 7.8 from page 129 in your text. So please again put this chapter to work on page 132, and please also take some time to retrace the learning path again on page 132 to have yourselves well prepared for your ensuing test. This has been your instructor, Scholar Huff, and thank you all for listening. Prepare well.